Well, hello again. So, when we last talked, we talked about the cell structure. In this case, we're looking at one part of that structure that's really, really important, the plasma membrane. Now, I know you probably looked at this with your holiday homework and the list of organelles. And in many respects, the plasma membrane is not really an organelle. It's only a cell part, and it wraps itself around the entire cell to enclose all those organelles. But really significantly important part of the cell. We always draw them in a 2D sort of way, but we need to think about cells as 3D objects. And of course, the plasma membrane wraps itself around in a 3D sense. So it's quite an amazing structure. So we're going to look at the structure, we're going to look at how that influences internal and external environments, and then how things manage to get across this structure that are needed, and how it sort of blocks out things that aren't needed. So, I'm sitting in the wrong place there, over top of the membrane. That's right. Membranes are made of two layers of phospholipids. So, we've got this part here called the head of the phospholipid, and these green bits are called tails. Embedded throughout are proteins. So, there's a glycoprotein here, protein and glucose mixture, a little carbohydrate side chain. It's a signaling location. We have proteins that are about movement through the cell. There's a big one here that I'm obscuring, and other proteins that are also part of that signaling ability, which we will talk about next year in Year 12. What we can't see here in this particular diagram, although these red things here might be an attempt, are the cholesterols that lock these two together. So we need to think about the membrane as two layers that are trying to get away from each other, because these heads are hydrophilic, and attract water, but the tails are both hydrophobic, and they repel water. So this is a lipid layer, bilipid layer. To move through this, you need to be lipid soluble or very, very small, hence the channels. But of course, if you've got two north ends of a magnet, they push apart. So these two things are alike, and they're pushing against each other. So the cholesterols are often used to help lock them together. And obviously these other structures help to achieve that too. Our cell is constantly repairing and monitoring these problems and trying to make sure that its fluid that's moving all the time, mosaic of all these other objects, stays together. And essentially it controls what comes and goes from the cell, and obviously it holds everything else together inside the cell. So, hydrophobic versus hydrophilic, these are sort of terms you need to be aware of. So if you're hydrophobic, you repel by water, or you repel water. So those tails are hydrophobic. If you're hydrophilic, you're attracted to water, or you attract water to you. So the heads tend to attract water and the water-soluble material inside the water to the cell. If you're polar, that's a molecule that has charged ends, so positive and negative ends. If you're non-polar, you don't have charged ends. And these are, again, really important considerations for how things can move across our membrane. The non-polar ones are probably more able to move freely against the polar ones, which can be repelled because of their charges. And size is important. Small molecules are really easy to move across. So oxygen is just two oxygen molecules, which are already small atoms, which are already small, joined together. So they're tiny, so they can diffuse through this membrane quite simply. A protein's masses of hundreds, if not thousands, of atoms, and they're far too big to come across a membrane, and they have to be broken down or come through a channel. So size is really important here. So we talk about external environments, and with a large organism like us, there's sort of two external environments. We've got an external environment outside our bodies, and we've got an external environment around our cells. That's the fluid between tissues and between cells. We call it extracellular fluid. That's external to the cell. And obviously the cytoplasm, or cytosol, is that internal environment that the organelles all sit in. It's important that it is fluid, because then all these things that we need to get into the cell are dissolved in that fluid and are able to move by diffusion or osmosis, or if need be, actually transported for certain diffusion, which we'll get to soon. And of course, cells need to have things like water and oxygen and glucose, amino acids to make their own proteins, lipids to make fats. and vitamins and minerals to enter the cell. So these things need to be able to dissolve into that fluid environment and move across by diffusion or osmosis. So this is our internal environment, of course, and you will be well aware of this now. So the cytosol is that material surrounding all of these organelles. So if we talk about cytoplasm, we mean the organelles and the cytosol. If we say cytosol, we're just talking about this internal environment where all this material has to end up and the cell makes use of it for all sorts of purposes. So how do you get things across a membrane? Well, membranes, therefore, are partially permeable. They allow some stuff through. And as I've already discussed, they allow small stuff, very small stuff, like oxygen and carbon dioxide, to cross. And this requires them to move by diffusion. Of course, they also allow things that are lipid soluble to move across by diffusion. So diffusion is a simple process. We can see in this diagram, high concentration of the dissolved stuff, the solute. That could be, say, salt. And there's none over here. So naturally, just happens by itself, no energy required, no decisions need to be made. Oops, my mouse is going in. This stuff here will start to move until we see a reasonably even distribution of salt in that whole space. If we put a membrane in there, of course, we can draw the salt across the membrane and create a concentration of salt inside. So it's a simple process, simply soluble material moving through the fluid from areas of high concentration to low concentration. Now, osmosis is a form of diffusion that relates essentially to water molecules or to the solvent that holds the solute. But we particularly talk about with water. So in this case, it's slightly different. So here we have water molecules, they're blue, and we have a high concentration of water molecules and a low concentration of the dissolved material. And we have a high concentration of dissolved material and a low concentration of water. Now, the water will move from here to here. It's not moving from a high concentration of water, though. It's moving from a low concentration of dissolved material, which seems a bit weird. But it's moving in relation to the dissolved stuff. It's the same effect, but it's a slightly different definition, and one I'd like to catch you on exams and things. And there are three important considerations around this. We talk about isotonic situations, hypertonic and hypertonic, and cells fall into all three. So what is isotonic? In an isotonic situation, you can see here that the free water, we've got the water, purple waters and pink, let's say sugars, they're basically an equal concentration. Now, that doesn't mean they don't move. They keep moving. So equilibrium doesn't mean dead still. It means things are moving, but the concentration of them all is remaining equal on both sides. So same amount of pink and purple in here to out there. They'll keep moving across, but their concentration is maintained. If a situation is hypertonic, in this case, we have less free water out here and more water in there, greater amounts of the dissolved solute, the sugar, outside to inside, then the water will look to move out. If the sugar is capable of moving in, you imagine, of course, it will move in by diffusion. Water out by osmosis, sugar in by diffusion, if it can cross that permeable membrane. And then hypotonic is the opposite. We've got lots of water on the outside, so that water will move in by osmosis, and lots of glucose, sugar on the inside, and if possible, it will leave. If it can't, it doesn't matter, the water will still come in, but obviously by diffusion, it would want to be leaving. We see this in operation in plant cells, and we've had a quite strange summer, but there's been some hot days, and plants in the garden will have wilted, and then you've watered them, and they've stood upright again. So the vacuole, that large vacuole inside the plant cell, has lost its water. It's moved out because the situation has become hypertonic. And then you've put water in the ground, and it's been drawn up by the roots, and those cells soak that water up again and become isotonic, of course, but from a hypertonic situation, from a hypotonic situation, lots of water outside the cells, moving in and filling the vacuoles. We also see this happening in fish. Fish, of course, live in water. Some live in salt water, some live in fresh water. Some, of course, are capable of adjusting their membranes to allow them to live in both, in those estuaries. And they rely on these movements of water and solute to actually keep their cells hydrated. But more of that later. So that's diffusion and osmosis. Sometimes you want something that's really, really big or not lipid soluble, and you need it to diffuse into the cell. Now, in this case, there's no energy being used. It's just a channel that exists for material to move through. So these use the protein channels. Oops, my mouse gone again. And we've got high concentration out here and a low concentration in there. And so that will move by diffusion, but it's facilitated because it needs this channel to move through, because it can't get through here. Either it's too big or it's not lipid soluble. And it's energy free, which is useful. Sometimes you need to spend energy to get the stuff you want. So you've got material outside the cell which you want inside the cell, but either it's too big to go through a channel, or too big to cross the membrane, not lipid soluble, so it can't cross the membrane. It may not have a channel for it, or you may just simply need heaps of it. You want to bring it against the concentration gradient, and you need to actually use some energy to draw that through. So again, it uses these channels, these protein pumps, and the help of a carrier protein, rather than simply a channel protein, and it moves these across from the outside into the inside. But this time it's cost energy. So active transport costs the cell energy. We also have the ability to move things by endocytosis, pinocytosis, and exocytosis, which is, again, an energy requiring process, but an adaptation of the membrane. So endocytosis, endo meaning internal, generally is about, and cytosis is a bit about cells, means we can move things in. So here's some solid material outside the cell, but we want it. And so the membrane wraps itself around that material and draws it into the cell. So we've got a little vesicle which contains this solid material. Pinocytosis is exactly the same. It's just that when the cell drinks, if you like, because these are fluid molecules rather than solid molecules, and does the same thing. Forms a little vesicle around it and brings it into the cell. And exocytosis is the opposite. We want to get rid of something. So the vesicle forms, contains whatever we're getting rid of. Often it's a protein we've made, and then it becomes part of the membrane and releases that material out of the cell. Done. And so am I. Next time.